Hi, my name is Kumar. As Bobby mentioned, I'm an associate professor in the radiology and diagnostic imaging department. I'm also an adjunct professor here in computer science. It's my pleasure to introduce Dr. Deepa Krishnasamy. She's a research fellow at the Brigham Women's Hospital, working with Dr. Andrei Fedorov in the surgical planning laboratory. She completed her PhD here at the University of Alberta in our lab under the supervision of Dr. Michelle Noga and me. And so currently she is focusing on developing publicly available and reproducible methods for enhancing lung cancer imaging. Additionally, she has developed and contributed to a 3D slicer plugin for multi-parametric MRI annotations of the prostate. Her research interests include prostate state data curation, deep learning approaches for MRI segmentation, and open source tools for visualization.
Thank you.
Yes, thank you.
Thanks. Okay, so hi everybody. Thank you for having me. First of all, I'm really excited to be back in Edmonton. Like super excited. Okay, so today I'll be talking about AI-derived annotations for lung cancer imaging collections, and this is using the National Cancer Institute Imaging Data Commons platform. So just a little bit of background. Kumar already told you I did my PhD here in radiology. And for the past 2 years, I've been a postdoc at Brigham Women's Hospital, working with Andrei Fedorov. And these are just some pictures of our team. And so I wanted to go into like what my lab's major interests are first, and then I'll go into the specifics of my project. So as many of us are working in AI, we probably know that reproducibility and transparency are major problems. This is with regards to the code that you develop, the data that you have and you want to share, or even the workflows that you develop, and your model weights, et cetera. This has been a problem for years, and there's been a number of approaches to try to rectify this problem. So for instance, model cards for model reporting, or different types of checklists, such as here, the claim checklist. And these are all great starts, but unfortunately, they don't cover all the problems that we have with reproducibility and transparency. So our lab really tries to focus on this. And we've been working on a number of different projects. So for instance, the first paper that we have here on the left talks about developing these workflows for the cloud. And my paper here on the right, this is with enhancing different lung cancer imaging collections, which I'll focus on today. And we also have prostate segmentation use cases as well. And for all of these, we try to provide like Google Collaboratory notebooks. We share all of our data and it's all in the cloud. And some of my other projects and interests, if you're interested in this, I work on 3D Slicer, which is an open source medical imaging platform. And for this, I've been developing some extensions for this. So like how to delineate the prostate using multiple types of MRI scans. I'm also working on segmentation and MR volumes, and also some data curation of prostate biopsy datasets. So let's start with the talk. So the first thing I wanted to talk about, what exactly is NCI Imaging Data Commons? That's going to be the first part of the talk. And then afterwards, I'll talk about what types of annotations that we actually generated using AI approaches. So let's start with Imaging Data Commons. So the idea of the data commons is that you want to have your storage and your data and any infrastructure that you have for computing all combined with each other. And once you do this, you can hopefully have visualization features or anything to be able to explore your data. And so many of you that probably work in the hospital do this the more traditional way. So you have data from your PACS system. You bring this data, you de-anonymize it, then you copy it to your local machine, you do all of your analysis locally, and then you try to share this with other people. But the problem with this is, you have lots of data copying going along. You really rely heavily on your local computer for your compute power. There could be many problems with security that's involved, and you really need some sort of big infrastructure. And so the cloud way of doing this is instead we're going to bring the researchers to the actual data. And this is great because the cloud already provides all the infrastructure that you need. And of course, you can also identify and choose what type of compute and storage requirements that you need for your particular use case. And so this is what we're going to be focusing on for the rest of the talk. So the NCI Imaging Data Commons is just one of many cloud-based repositories for cancer imaging data. And this is actually a component within a larger data commons, and this is called the Cancer Research Data Commons. And in this you have, as you can see here, like genomic data commons, proteomic data commons, and many other areas. But this, we're just going to focus on the imaging part. And so what do these data commons actually hold? So we have about 45 terabytes of different sorts of imaging data. This comes from radiology, digital pathology, and many others. We also have annotations that are also derived. So for instance, maybe we have volumetric regions of interest, so different segmentations. Also on a slice level, we have different types of features available for you, including landmarks and any type of analysis result. But most importantly, we also include clinical data. So for instance, maybe you need patient demographic data, maybe you need to know the survival of the patient, if they had cancer or not, or what type of cancer. So that's just a basic introduction. And you can see over the years, this has been going on for about 4 years now, that the number of data sets that we've been including in the NCI Imaging Data Commons has increased with regards to the size and number of collections. So you might be wondering, though, what type of format do we actually store all this data in? So maybe some of you have heard of the DICOM format. And this is a pretty standard format that we have in medical imaging, and it's been used for over 30 years. And the great part about DICOM is that you can actually use it for harmonization across many different vendors or hospitals. And you've probably used it in many applications. And the great part is that you can actually use it in commercial or open source applications as well. And David Clooney, who's one of the main editors of DICOM, is actually part of our team. And so he really helps us to make sure that we understand how to use the standard properly. But most importantly, there's no other alternatives that can match what DICOM has to offer. So for instance, say if you need information about the orientation of a patient, or you need to know how the patient was scanned, a lot of these other formats remove all that information. But we decide we want to keep that information so later to use that to form your cohorts for AI analysis workflows. Okay, so now that you have this data in this data commons and you have some cloud resources, what can you actually do with all of this? So the first thing you probably want to do is you just want to explore your data. You want to see what types of data there are, what modalities, what segmentations are available. After this, you want to just choose a specific subset of that data. So what fits your particular AI workflow needs. After that, you want to analyze it, apply your algorithm to it, and then hopefully you would like to share your data in a reproducible manner. So let's talk a little bit about the exploration. So I'm going to go to the actual portal and give you an idea of what this looks like. So, okay, so this is the Imaging Data Commons portal. So what I'm going to do is I'm just going to click on the images and we can see what types of data are inside. So here there's different filters depending on what you're interested in. So for instance, maybe you want to look at particular areas of interest, like the chest, if you're looking at lung cancer. Once you do this, you can then click on different collections as seen here. So for instance, I will click on this particular one, which has to do with lung cancer, and then you can click on a patient, and then eventually you can view all of your data. So if I open this up here, this opens up an integrated viewer called OHIF. And now this particular patient only, or this particular study just has a single series, but ideally we can look through all the different sorts of data sets that are involved. And I guess it's taking a little bit of time to load this up. Okay, well, you can imagine that we have some data in there. So let's go back to the slideshow. Okay, so here's a little video that we took of some examples of radiology images that we have within this portal. So here you can see the different segmentations as you scroll through all the slices. They're in different colors. So here we have like the heart and aorta, trachea, and esophagus. And you can store these in different types of formats, so segmentation objects or ones to do with radiotherapy. And this is just an idea of the type of data that we have. And also you can explore this data using a dashboard. This is also part of the Google Cloud Platform, which is what IDC uses. And here you can search by different modalities or manufacturers. And so hopefully all of these can help you actually form the cohort that you want and are interested in. So after you've explored your data, now you probably need to choose a specific set of the data that you're interested in. And so you could do this by the portal, going through and clicking all those filters that I showed you before, but this is not really efficient. Ideally, you want to look at the actual DICOM data. So what happens is you have all this DICOM data and we can extract different types of metadata from that. So for instance, like what type of modality is it? How many slices are in here? What's the pixel spacing? And each of the X, Y, and Z directions. And you can use all of this information to then filter and query out the data set that you need. So this is like an SQL query that basically you could write. Maybe you want to get rid of like geometrical inconsistencies, for instance. So after now you've subsetted your data, now you're ready to analyze it and apply your algorithm to the data set that you found. So this is the first part of now I'm going to talk about what are the first steps that we've done with actually generating these AI-derived annotations. So our overall goal is to enrich some of the CT collections that we see in IDC. We also have MRI and digital pathology, but there's some other people working on those, so we're going to be focusing on CT collections. At the same time, we want to make sure that we can understand and compare different state-of-the-art tools that have been developed recently. Along the way, we want to make sure that we can provide you with different recommendations for how to make this all reproducible and transparent, and then hopefully keep the cost of the cloud down as well. So just to give you a little preview of the type of data that we've actually generated in this first step. So here in the portal, you can actually filter on the left by different analysis results. So if you see here, we have something called NNUNet BPR annotations. And basically, if I click this link, which I'll show you later on in the presentation, this is the type of data and annotations that we've come up with. And so we've used currently publicly accessible AI models, and we've been able to generate different volumetric segmentations as shown here, and also different slice level annotations. So now we'll go into actually how we develop this. So one of the largest imaging collections that is in IDC is the National Lung Screening Trial. And this was a study that was performed over 33 different centers around the US. And this was basically scanned for people that had a high risk of lung cancer. And you can see that there were over 26,000 patients in this collection. So this is the largest collection, but unfortunately, none of the data sets or any of the patients have any labels. And this is really important because you need to know where the tumor is. Maybe you also need to know which organs that shouldn't be affected when you do radiotherapy, all sorts of different annotations that could be useful. So this is what we decided to focus on in our first project. Additionally, we're also missing a lot of other metadata that could be important when you want to learn about what type of data you actually have. So for instance, there's just one particular tag called body part examined. This basically just tells you, okay, what part of the body was the scan actually looking at? So most of them have the chest, which is expected because lung cancer has the largest amount of data in this particular collection. But we can see that the second largest is null. And this is not really useful because obviously this is incorrect. We also have tags that are missing for different patients. And ideally, we also need this on a slice level, not on an actual patient level. So are there ways that we can use to actually input and generate these annotations that would be needed? So for instance, if you wanted to look at just patients with lung cancer, maybe you need to find patients that only have a chest scanned. So we needed to have some criteria before we actually started this whole annotation process. So this was some of the criteria that we came up with. So first, we wanted to make sure that there's a peer-reviewed publication, that there's an evaluation that's publicly available of the performance. And sorry. Okay. We also want to make sure that there's sorry about that. Okay. We also want to make sure that it's performed well compared to other algorithms. And of course, we wanted to make sure that's also open. Oh my gosh. Sorry, it's also open source in an ideal case. Okay, so the first task that we came up with was thoracic organ segment. Sorry about that. Okay, thoracic organ segmentation in CT. So we use the popular NNUNet method, which has been shown to perform very well in many different cases. So we chose a particular pre-trained model that works for radiotherapy cases. So here we can see that we have segmentations of the heart, the aorta, trachea, and esophagus. And this is proven to win a number of different challenges as well. And the second the second algorithm that we chose was a per slice body part annotation in CT. And this was also developed by KFC. In the ideal case, what we want to see is here we have different landmarks that have been annotated by this method. So for instance, we have the lung up to the bottom part of the lung and the top part of the lung. And ideally, you could use this to then subset your segmentation, maybe get rid of some false positives downstream in your downstream analysis. And so what's the actual workflow that we have that's enabled by Imaging Data Commons? So as I showed you before, we first want to make sure that we can query our data from our cohort that we actually are interested in, interested in analyzing. After this, we'll take one of our models and we'll perform inference, and then we'll obtain all of our results in standardized format. These could be segmentations, these could also be different slice level annotations, or also we could extract features from each of these segmentations, such as volume or mean intensity. And then ideally, we'll then visualize all these results on public data. And so how did we actually do all this analysis? So most of you have probably heard of Google Collaboratory Notebooks. And this works really well if we don't have a lot of patients. So we started off with a free tier. We developed a minimum working example. Now this was good, but we decided we needed this to run a little bit faster. Okay, so let's go to Pro. That's a little bit better. But unfortunately, since we're analyzing hundreds of patients, this really would take forever. So we decided, okay, let's design a custom GCE virtual machine and do our configuration that way. And as I said, that collection that we were looking at actually had 26,000 patients. So this is a diagram that you might see in a typical paper, which says which patients were removed from the analysis and for what reasons. Now, this is helpful. And for us, we decided to look at just the patients that have actually tested positive for cancer. But to actually come up with this number, we developed a query, as I showed you before. And you're probably wondering how much all of this analysis actually cost. And this cost about $300. But as I'll show you later on in the presentation, there are actually much more efficient ways of doing this analysis. Okay, so let's come back to here where I can actually show you some of the results of what this looks like. So here I'm opening up the analysis results. We looked at two collections in particular, and now I'll just open up the viewer. Okay, so right now we have the CT scan that's being loaded. And if I scroll down here, you can see the different types of objects that we have for this particular scan. So we've created segmentation objects, which hold the volumetric measurements, and which I'll show you right here. So if I click on one of these, takes a little bit of time to load up. Usually it's a little bit faster. Okay, we'll wait for that to load up and I'll come back to that later. Okay. Another way that we can interact with this data is by actually looking at a dashboard. So as I said, we actually extracted different types of features from each of these regions, such as volume, mean intensity, and different shape features. Now, the problem is, since we did this over hundreds of patients, you have no way of knowing if your analysis is actually correct. And if you don't have some sort of expert like a radiologist to sit down and look through all the annotations with you, then we needed some quick ways to quickly figure out if there's any outliers. So one of the things that we tried to do was just pick different regions, pick a feature that we were interested in plotting, and then create a plot like this and look at values that either were very low or very high. And then hopefully we could then take these to the expert radiologists and see if they could interpret why there were problems with maybe the method that we chose, or maybe there was a problem with the data itself. And this is all done in the Google Cloud platform. Okay, so finally we're up to the sharing of results. So the first thing that we did was we uploaded all of our results onto Zenodo. And then from here, these were all imported into the Imaging Data Commons platform. And after this, we created a GitHub page where we included all the notebooks that we used to actually create all these files and do all the generation. And we included also notebooks to show you how to actually use this data directly from IDC itself. So for instance, if you wanted to download the data, you want to visualize it, maybe you want to subset it more somehow, we show you how to do all of that. And we have a paper that goes through all of these in detail. And so as a summary, we've made all of our data and AI annotations available by uploading them to Zenodo and into the repository itself. And we've shown you notebooks how to actually interact with this. So this is great, but we only analyzed maybe 1,000 patients or so. So what happens now when we want to actually scale this up to all 26,000 patients in that particular NLST collection? So this is where I'll talk about the next part, how we actually scale it up. So what we decided to do was we could have used those two previous AI models that I showed you in the past. But ideally, we really want segmentations of much more organs and muscles. So there's this popular method that's also based on NNUNet, and this is called total segmentator. So this has been developed for CT images, and you can get a segmentation of 104 different anatomical structures. And this has already been shown to be pretty robust for different types of CT scans. So we thought, what if we apply this to the entire NLST collection and then make all of our data available to everybody? And hopefully, once they have all these segmentations, they can then use that to infer different things about the patient and perform their own clinical studies. So this follows the same general pipeline that I kind of showed you in the past. So we have our data, we want to first filter it, make sure we get rid of any geometrical inconsistencies or maybe scans that are incomplete. We run our model, and then eventually we convert all of these to diagram objects, as I said before. So this gives you an example of what this data actually looks like. Here you can see an axial scan, you can see a coronal scan here, which is a scan from the front, and a 3D rendering of what this looks like in the 3D Slicer open source software. So this is very impressive, and we were able to run this on 26,000 patients. But if we did this one at a time, it would have taken over 785 days. So instead, what one of my coworkers did was he used a program called Terra, which basically you can do distributed computing. So he was able to create and run virtual machines that ran in parallel. And by doing this, we actually only needed 8 hours and 7 minutes to run over 26,000 patients. And that's incredible. And it only cost $800. So this was some great work that was done by my coworker, and hopefully we'll have some papers coming out so you can read more about how we did that. But what I wanted to focus on more is not actually how we did that analysis, but what can we do after we get all the segmentations? Because how can we possibly go through 26,000 patients and see if there's any issues? Maybe the AI model didn't work well. Maybe a patient had some sort of like inconsistencies or tumors, anything like that. So we really wanted to spend a lot of time trying to come up with different heuristics to understand our data. And then once we could filter out the data that was actually useful, then we could take these to a radiologist and say, okay, now we want to make sure that these are correct, maybe perform some population studies and compare against literature. So that was our overall goal. But what I just wanted to show you, if it works, I'll show you a quick link of what this data looks like, because I think it's pretty impressive. So yeah, here we see, so what I'm doing is I'm scrolling down like through the axial view. And here you can see, this is probably not 104 segments because the whole body wasn't scanned. But yeah, this is the type of visualization that once we get this data back into the repository, you'll be able to see. So I think that's pretty impressive. And so now what I'll focus on for the next few minutes is how we do this quality assessment. And as I said, we want to make sure we're not replacing the expert or anything. We just want to use them efficiently. So what we did was we decided to try to develop a number of different heuristics. So are there ways that we could figure out if patients don't conform or certain regions in the body don't conform? So we thought, okay, let's divide this by either a single region or let's look at multi-regions at the same time. So for instance, we want to make sure, say we're analyzing the volume of the liver. We don't want to analyze patients where the liver has actually been cut off because the scan ended. We want to make sure it's fully segmented. We also looked at many things to do with the vertebra because we have all the vertebra separate segmentations in this particular model. We also want to do some outlier detection. And so I'll go into these and how we use these to filter out some cases. And what I also wanted to make sure that everyone understood is that, we're not taking all this data, converting them to like 3D NumPy arrays and then doing all of our analysis. Like that would take a really long time. So what we're doing is we're taking all these diagram objects and then we're extracting the metadata that we need from all of them and using SQL, like I showed you before, to filter. So this is really fast. But most importantly, if you have your data all in the same way, you can do the same exact things that we just did. So now I'll show you some examples of some of the heuristics that we developed. So first we want to make sure, is a region fully segmented? As I said, you really need this before you want to look at any further analysis of your patient. So here we see three views of the CT scan. And here's an example of one of the organs. And you can kind of see at the bottom that, yes, this organ is cut off. Okay, so we can detect that. Now you filtered out for those patients. Now you want to make sure, since we're looking at lung cancer imaging patients, it's very likely that the vertebra will appear in all the slices. But unfortunately, because the model's not perfect, of course, the vertebra segmentations may be wrong. So this is an ideal case that you see in blue, where we have just a single vertebra segmentation. But now if you look in pink and yellow, we can see that those clearly are not the two vertebrae. So can we detect these cases and then remove these patients or flag them so then a radiologist can look at them? After we do all of this, maybe we also want to look at the volume of all of these. And then maybe we want to filter for some of the outliers that we get. And these could come from a number of other previous issues. So now that we've done the single-based region, we can look at the multi-region based. And as I said, since we're looking at just on lung cancer, the vertebra will likely be present on every single slice as seen here. And we want to make sure, of course, that the order of the vertebra is correct. So is it going from cervical to thoracic to lumbar? So those are just some of the tests that we came up with. And after all of this, what we want to do is actually compare to the population and compare our results to a paper. So this is an example of what we came up with. Okay, so we're plotting just the volume of each of the vertebrae. Here we're going from like the topmost of the body to the bottom. And after all of our filtering of those 5 heuristics that I described, we actually get rid of about, we have a 43% data loss, which might seem like a lot, but there's really a lot of patients that should have been actually removed in this case. And so what we decided to do is we found a paper that was similar to our analysis that looked at volumetric analysis of the thoracic and lumbar vertebral bodies. And we tried to all compare our results to that. So here what we're looking at is we are looking at the volume of the different vertebrae across the x-axis with the volume in the y-axis, and we're comparing the males versus females. So males in blue, females in orange. And we can see that we have the paper results here at the bottom, and then our results here in the dotted lines at the top. And of course, there's a major difference between the two sets, and this is because we're actually looking at different parts of the spine. So the paper just focused on the central vertebral body, while ours actually segmented the entire part of the vertebra. And this is because of how the model was actually developed. But we can see that that's okay because our results actually follow the trends of the paper. And that males have a smaller volume, males have a larger volume than females, which holds for both cases. And that there's a slight drop off when you get to some of the lower vertebrae here. So this is just one possible example of some of the analysis that you can do if you want to compare to literature. And we've developed, as I've showed you before, a number of different like dashboards, so you can interact with all this data. You could filter by different manufacturers or gender. And I just wanted to show you a quick example of what this looks like. So say for instance, you want to choose a different manufacturer. You can choose different kernels because we're looking at CT data. Again, you can filter by gender, age, or race. And this is all data that we have because we have the clinical data also associated with this particular data set. Anyway, so I've provided all these links so you can explore and have fun with this data dashboard. Okay, so in summary, I've been able to show you how we've been able to annotate different lung cancer imaging data sets, and hopefully I've provided you with some recommendations so all of the code and data that you have and want to develop, you can make it reproducible. And hopefully I've also offered you some solutions for using the cloud for your analysis, or at least giving you some inspiration. And that's it. So thank you.
Thank you for the wonderful presentation. Let's open the floor for questions. Let's get a couple of them while we wait for the audience. One is the data privacy. So how do you, what is your recommendation on, or how did you handle the data privacy? Because this usually the medical data is sensitive, so what is the typical process on that?
Yeah, sorry, I think I probably didn't explain this well, but actually all of our data is public. And so it comes from, most of it comes from a different repository called the Cancer Imaging Archive. And there they already have all the data de-anonymized. So they've already taken care of that, and then we ingest that directly. And then we made sure that any other place also has gone through their own anonymization process. So like we wouldn't take care of that on our side.
Okay.
Yeah.
So let's say our team wanted to contribute some data to this repository. Is it something possible, or if we follow the proper protocol of getting the approvals and everything?
Yeah, I think it is possible. So like one way you could actually submit your data to TCIA after you've done your anonymization, and then we would pull it from there. But say for some reason that they don't accept it, then maybe we could work with you. And you show us that like this is a promising algorithm, this is a promising data set, then I think that could work.
Okay.
So
and all the data set that's already there, it's available to any researchers who will.
it is, yeah. And you don't even have to use the cloud for any of it. You can just, if you want, you can just download it to your own computer. So,
okay.
Yeah, for free. So.
Yeah, it's a nice presentation. I have a question about the AI-based tools that you use for annotations. Are they expert informed? Because you mentioned that you're building these systems not to really replace an expert, but to help them function better. And when you start performing these annotations, which is like the first step to get data into a model, does your framework take into account expert knowledge in formulating descriptors for these annotations, so that right at that point you get informed annotations, like domain-specific annotations?
Yeah, so I think so for right now, we actually are just taking the pre-trained models and we're applying them to our data set. So yeah, unfortunately, we're not doing like any further analysis of that,
So the pre-trained model gets you the annotations?
So like we take a pre-trained model that like say gives you annotations of those 4 regions, and then we do inference on all of our CT data volumes.
Oh, okay.
Yeah.
And my second question, how easy is it to extend this framework to any other sort of data and other sort of data formats that people from other disciplines can have? Or is it just limited to medical imaging data alone?
Yeah, that's a good question. So I think DICOM, itself has been used in other areas outside of medical imaging, or at least, apart from CT, it's been used for like pathology and MRI and everything. But yeah, if you have something that's totally outside of the medical imaging part, I'm actually not sure. Yeah, if that would work so well. Yeah, that's a great question, but we can talk about that later. Right.
Thank you.
Yep.
Thank you for a great presentation. So I have 2 questions. One is, how do you check the quality of data before even starting the annotation? How do you filter?
Yeah. So I think if you remember, I showed you like how we can write SQL queries. So what that's actually doing is we have metadata from these DICOM files saying like how large is your data, like what are your dimensions, what's your voxel spacing, what's the orientation of your patient. And if you could write a query to just filter out those different fields, say like, oh, I want patients that only have over 100 slices in them. And maybe this would get rid of like incomplete slices or something like that. So basically we had to come up with like all these different criteria of what we thought would create a good data set. Yeah.
And also it only accepts the DICOM format, or it also works for other formats?
Yeah, so actually, everything is dependent right now on the DICOM format. I guess if you had like Nifty or other like formats, you could actually convert to DICOM and do the same exact thing. The issue is that extraction of the metadata, that only comes from being in a DICOM format. So yeah, but I think the rest you could just do with like Nifty and stuff. Yeah.
Okay, thank you so much.
Thank you.